As we move into Chapter 9, we are back into discussions of techniques that are used in the laboratory to obtain information. We previously discussed working with proteins, but unfortunately, sometimes when you start with proteins in your laboratory work, you end up only getting very small amounts of the protein that you are attempting to purify and study. And when working with enzymes, typically some of the purification methods lead to denaturation of the enzyme. So what we do a lot of time in the lab is we'll work with DNA. Now we can work with DNA to simply amplify the DNA itself and do studies with DNA. But we can also take that DNA that we have amplified and began to study and then use an expression vector, produce protein or enzyme to study itself. This is the keys that we use to study a lot of diseases, and to study whether those diseases are inherited or not, we start on the DNA level. So let's jump right in and talk about some of the techniques. One of the most important things we do when studying DNA is DNA cloning. And on this slide, I just show you the general scheme. So what we're going to do is cut out a small piece of the DNA we're interested in studying, shown here, from the larger chromosome, and then that is going to be inserted into a vector. The vector is a plasmid or anything that is suitable for carrying the DNA. Then using DNA ligates, we will ligate or combine the small piece of DNA we're interested in studying with the circular piece of vector DNA. This is now a recombinant vector. The recombinant vector is then inserted into a host cell. The host cell is there to simply grow and produce much more of this plasmid and therefore the DNA of interest as we continue. Now, this is a very simplified version of DNA cloning. Since I've started using some of these terms, let's make sure we're comfortable with a couple of the terms. Recombinant DNA is the term we use anytime we have taken a piece of DNA from one source and combined it with sequences from some other source. It has to be an artificially created piece of DNA. Cloning, the word by itself, simply means making identical copies. DNA cloning is making copies of a particular gene. That gene is typically isolated from one organism that serves as our source organism that we are trying to study, and then we're amplifying that target gene within another organism. DNA cloning is what's on this slide that I was demonstrating to you. So let's jump right in and look at some of the details of how we can make this DNA cloning happen. The first step we saw in our cloning scheme was cutting the gene of interest out of the chromosomal DNA. We do this with a group of enzymes known as restriction endonucleases. These endonucleases specifically cleave the phosphodiester bond between two nucleotides. They are very specific. They were originally found in bacteria, and bacteria use them to get rid of any odd DNA that's not supposed to end up in them. But what we do is we've studied these restriction endonucleases, so we know this particular endonuclease is going to cleave at these particular spots. This picture I've shown here is the type of endonuclease we use the most. It produces these sticky ends where you end up with small pieces of DNA that overhang. The advantage to using these sticky ends is when we try to reanneal the DNA back together, well, it allows for better binding and it's more specific. If you cut straight down, for example here, you would end up with a blunt end. And that works, but you end up with a lot more error that you have to sort through when you're trying to do your studies. So what we would do in the lab is we would cut the DNA with these restriction enzymes, then we would run a gel or somehow isolate that DNA. Once we have the DNA isolated, we can insert the DNA into a vector. One of the most common cloning vectors is a plasmid. Plasmids are going to be natural, small circular pieces of DNA that come from bacteria. They are very good vectors because they do not require insertion into the bacterial chromosome. They can replicate outside within the bacteria, but outside of that chromosome, and they can amplify very quickly. Cloning vectors typically have some restriction endonuclease sites shown here so that we would know exactly where this vector would be cut by enzymes. It's also going to have some resistance genes. That way we can select and make sure we're only growing organisms that have had the cloning vector inserted. Once we have cut our gene with our restriction endonuclease, we also will cut the vector with the same restriction endonuclease. This allows these sticky ends to match up. So once we allow these small fragments of DNA to exposure with this DNA vector, then we can add the DNA ligase into the mix to form our recombinant vector. There are multiple types of DNA ligase. One of the most common used in the lab is the bacterial DNA ligase because it only requires NAD for function of the enzyme. Now that we have our gene of interest inserted into our plasmid, we move into antibiotic selection. So we've got our piece of DNA that we're interested in inside of our vector. We've transformed it or put it into the bacteria or whatever organism we're going to use to grow, and now we move into antibiotic selection. Okay, so just from the beginning again, we take our vector, cut it, we cut the DNA the same way, ligate it all together, then the cells are transformed. Now I've used the word transformed a few times. Let me make sure that you understand what we're talking about at that point. Transformation is the kind of fancy way of saying inserting the vector into the host cell. Transformation requires a process of heat shock as well as calcium chloride treatment. The heat shock and calcium chloride decreases the integrity of the cell wall and the membrane of our host cell so that they're more likely to take in that vector of interest. Okay. So you can see from this little diagram what we've done is the ampicillin resistant gene, that's where we cut and inserted our DNA. So the vectors, the recombinant DNA that we have that has our gene of interest, the ampicillin resistant gene has been disrupted. So we can then grow our bacteria containing our vector on plates that have just tetracycline, which everything should grow. And then we should have a plate that has the ampicillin and the tetracycline. And only I think I may have said that backwards. A bacteria that have had the ampicillin resistant gene blocked are not going to be able to grow here because they're not ampicillin resistant. Now that we feel like we've completed our process, we're then going to separate our DNA by electrophoresis. So the book kind of leaves a few little steps out. We have our carrier organism with our recombinant DNA. We let it grow, and then we purify our DNA. We can then place the DNA in an agarose gel. Since DNA is negatively charged, it will naturally move based simply off of really size. The smaller molecules are going to move faster through the agarose than the larger molecules. What we usually do with this are restriction digest maps. That allows us to, of course, make sure we've purified some DNA, but we can also test and make sure that we've inserted the plasmid in the correct orientation. Because if you think about it, if you just have those two sticky ends, that piece of DNA could insert in either direction. So we cut it and we know exactly what size our pieces should be if it's in the correct direction. Now all of that is just expressing a piece of DNA within a plasmid for the purpose of producing more DNA for DNA studies. If we actually want to express the DNA and make a protein product, we use a slightly different type of vector called an expression vector. Expression vectors are going to have all of these items here. Promoter and operator sequences which allow us to initiate expression, which would be transcription, translation. We have the code for a ribosome binding site. So whenever we produce the mRNA, it will bind to a ribosome so we can allow the translation to occur. We also have transcription termination sequences that tell it, okay, you've transcribed all of the DNA that we're interested in. And then also, really important is the polylinker. This is the piece of the expression vector that we know exactly where the restriction endonuclease sites are found. So we can cut and insert our DNA where we would like. There's lots of different expression vector systems. Of course, bacteria are really easy to use, very well understood, and they grow very fast. But if you are interested in studying any type of protein that has a modification, such as a amino terminal modification or glycosylation, then you're going to need to use the mammalian cells. Now, they're more complex, a little harder to grow, but it just depends on the type of protein you're interested in studying. So here's a picture of a typical expression vector, and you can see we have the promoter and operator sequences. They always have this little arrow showing you this is where transcription would begin. This polylinker region, when you're working in the lab, you would get a nice blown up picture of it so that you could see the exact restriction endonuclease sites. And then transcription termination sequence is right after your polylinker. You're going to have a antibiotic resistance gene, so you can do the same thing we saw in that last figure, where only the bacteria or the mammalian cell, whichever you're using, that contain your expression vector will be able to grow. Something not shown in this picture that we will often see in an expression vector that we'll talk about in just a minute is somewhere right in this region. Marker's not working, but somewhere right in that region, right before the transcription termination sequence, you would have a marker that allows purification. So that purification marker would become part of your mRNA and therefore part of your protein. So it makes easier purification. One thing that we can do at this point in our studies is site-directed mutagenesis. We can alter the DNA one nucleotide at a time. That will lead to an altered code, which will alter the protein that we produce. One of the easiest ways to do this site-directed mutagenesis is to produce a small little primer that can bind to our vector, and it's going to match up enough with the DNA sequence that it will anneal, but we make it where it's got one bad spot or one nucleotide that's been changed. So if we add some DNA polymerase, we're going to continue producing a complement to our plasmid, our vector. Then we can get rid of this original strain by having it glycosylated or something, methylated, something like that. Then more polymerase, we can end up with the piece of DNA that has the mutation that we're interested in. And once we put it inside the cells for our expression, all these little nicks will be fixed. So whether we mutate it or not, we're ultimately going to need to make our protein. So we'll induce these cells, tell them, okay, time to make protein, then we'll purify. If we have tagged that expression vector with one of these really common tags, we know exactly how to purify our protein. The two that I've actually used in the past are adding a polyhistidine tail. The polyhistidine tail is very easy to remove. It's always very obvious at the carboxy terminus, and it binds to a nickel column very well. The glutathione S transferase tag or GST tag is also very common. It binds to a glutathione tag column. So let me show you a little bit about what this looks like. So here's our gene of interest, right? When we put it in the expression vector, we add the gene for GST. So when we produce our protein, we've got our gene we're interested in fused with this tagging protein. Put it all in the cells. What we make are the protein with the tag. So if we add it to a column that has the glutathione on it, only our protein is going to bind to this tag. and then we can elute specifically by breaking that bond. And we now have a very pure fraction containing our protein, but our protein does have that tag on it. You have to be a little bit careful if you're working with enzymes. You want to make sure that the tag you have does not mess up the fold of your enzyme so that it's not functional anymore. You also want to make sure that if you need to remove that tag, it's easily removable. So just things you have to think about and work out when you're working in the lab. Another process, just to review with you, that I'm sure most of you have heard of or probably done at some point in your undergraduate career, is polymerase chain reaction, or PCR. This is a fast and convenient way to mix some stuff up and walk away and amplify your DNA in a very large quantity. Okay? So in order to do this, you have to have your piece of DNA you're interested in, of course. Lots of nucleotides to use to add. You have to have a primer. You design that primer so that it will specifically bind around your gene of interest, and the polymerase will only amplify your gene of interest. And then probably the most important is your thermostable DNA polymerase. You can't add just a regular DNA polymerase because we're going to heat to such high temperatures. And if you think about it, the reason we're heating to the very high temperature, so first cycle is heat, we want to separate our two strands of DNA. We learned in our last chapter when we reviewed the basic chemistry of DNA that one of the easiest ways to denature or separate the two strands was heating. And if you heat to just the right amount, when you cool it back down, you will have annealing. The two strands will come back together. So it's a very simple process. We separate the strands. The primer specifically binds around our gene of interest. We then allow the polymerase to fill in the gaps, elongate our DNA, of course, 5' to 3' like always. Then we keep repeating that over and over and over again. Now we have 2 more strands. Heat it up, separate them, add the primers, cool it down, allow the polymerase to work, continually amplifying our DNA. That's basic PCR. We can use PCR for several other things. We can do reverse transcriptase PCR, where we actually take mRNA and use that as a template. So we just have mRNA in there, and then the reverse transcriptase will add the nucleotides instead of the polymerase. This is how we make cDNA libraries, which we're going to talk about in just a minute. There's another form of PCR called quantitative PCR. Quantitative PCR just allows us to actually measure how much of the gene we have. So it allows you to develop a ratio so that you can tell how much you had to begin with based off of the exponential phase of the growth. Now, as we start to continue thinking about this reverse transcriptase, or RT-PCR, we think about, why would we want to do that? Why would we want to amplify from a piece of mRNA instead of DNA directly? Well, if you can remember back to your undergraduate days, something we'll actually go through in great detail in biochemistry too, whenever DNA is transcribed into mRNA, that's when we get rid of all of the non-coding regions. We only have the coding regions, the exons, the parts that are actually expressed. So it's not always, we don't always want to amplify an entire genome, all the DNA. We may want to just amplify the pieces of the DNA that actually produce protein. And that's what we're doing when we're constructing a cDNA library. To make the cDNA library, you purify all of your mRNA by using the fact that all mRNA has a poly A tail. You can then take that purified mRNA from an entire organism or an entire body part. I've done it with mouse brain before. And then you do the RT-PCR. You use the reverse transcriptase. You make DNA from every piece of mRNA. Once you do that, then you've got your DNA template, and you do normal PCR to get lots of copies of the coding regions of your DNA. Another topic that we can look at just a little bit is using DNA to localize, using DNA techniques to localize where a protein or a piece of DNA is functioning. Now this is really neat. We can do this within single cells, that's what's shown here, or we can do it within a whole organism. This is a worm. And what we would do is we would take the piece of DNA we're interested in and produce a recombinant piece of DNA that has a protein tag to it for fluorescence. Then just insert that into the organism, see where it binds, and then it's going to fluoresce in the area where the DNA is located. You can do something similar called immunofluorescence. So it has an advantage that you don't have to add anything to the expression. You just have to produce these antibodies that can specifically bind to your protein or DNA of interest. That's what's shown here in yellow. And then what's in purple is the secondary antibody that binds only to the primary antibody, and that's what can fluoresce. I did part of this in my doctorate research, and I localized where the enzyme I was interested in was found in the developmental stages of a fruit fly. And it allowed us to prove that the enzyme was working within the secretory vesicles, so further providing evidence that the enzyme I was working with was a post-translational modifying enzyme that had never been discovered in the fruit fly. All right, so let's spend the last few minutes of our discussion talking about some of the newer technologies. Now, these are extremely advanced. It's not something that I would expect you to be able to repeat details of. And now what we'll go over in class on Tuesday is what I would expect you to do with the information that we just finished covering. And I do expect you to understand it in enough detail that you could use that. But these newer technologies are what's allowed us to study thousands of genes at once. It's allowed us to really just do large screenings. And those of you that are interested in the medical field should really find some interest in this, because we're now able to screen for inherited diseases, which is absolutely amazing compared to what we were able to do just 10, 20 years ago. So all of this started with the development of microarray chips, and what these are, they allow us to determine patterns of expression. So I think it might be a little bit easier if we go to a picture. So what we do is we take this plate, okay, and then we can take, in this example, they've got a fertilized egg and a tadpole. So just two different stages of growth. They get rid of all that. They purify just the mRNA, make cDNA libraries, and then they anneal all of this to a plate. So you end up with your microarray, okay? And then what you can do is expose other things to your microarray now that you've produced it. Now all of this is done through high throughput screening. It is not someone sitting there pipetting something into each one of these little wells. But once we produce the microarray chip, we now have a screenshot of every piece of DNA that's found in this fertilized egg, as well as every piece of DNA that's found in this tadpole. So let's say we now wanted to look and see, all right, a particular protein. Is it expressed only during the fertilized egg stage, or is it produced in the tadpole stage? We could then take that protein of interest, figure out what the DNA sequence is to make that protein of interest, and then apply it to the entire microarray chip. Okay? And then the different colors tell us whether expression occurred or not. We get the colors from also adding in some fluorescence markers. So it's a very complex process, but what I want you to understand is it allows us to take a one-shot look at everything that's going on within that one organism or developmental stage. One of the most interesting things we've done with this is figure out, we've done genome-wide genotyping, so we know which genes are present in individuals. So if those are altered genes, we know a disease is present or likely to be present. We've done this with fertilized eggs that are part of in vitro studies, so that people can actually select a fertilized egg for implantation that does not carry a disease. We also do this so that we can pick the sex of the baby. All right. So lots of interesting things that we can do with that. We also now have the Human Genome Project, where we have sequenced the entire human genome. And this allows us to do all sorts of things. We can make phylogenetic trees. From the Human Genome Project, we have figured out what DNA is specifically tagged for in our bodies. So we know how much of our genome is responsible for producing protein. We figured out we don't know what a lot of our DNA is for, and that's led to a lot more in-depth studies. So to show you one thing we've learned from the Human Genome Project, this is looking at just the protein coding genes. We know that 16.2% of our protein coding genes code for enzymes. And you can just kind of look at the picture, but I think this is pretty impressive. Even though we know that all of this we're looking at codes for proteins, we don't know what 37% of the proteins even do. We just know we make them. Now that's just the protein coding genes. When we think about the DNA, then we have no idea what it's for. We figured out that half of our human genome is made up of these repeated sequences that can hop from one place to another, and they're called transposons. Most people believe those are related to evolution. 25% of the human genome consists of sequences that are just in between the protein coding regions. Don't really know the function of that. And then 3% are these simple sequence repeats, the SSRs. You'll see a lot of those. That's a really new hot topic. They have, we've reported links of that to metabolism as well as production of telomeres. And with the telomeres, that's related to aging of cells. It's all sorts of interesting things. One thing we can do with those studies is look at the single nucleotide polymorphisms. And again, this is something we could spend all day talking about, but just to give you an idea of what we can do with it, we can take several individuals and look at a particular DNA region. And if we notice we've got lots of change in the nucleotide sequence in particular areas, if a lot of changes are within a small region, that produces a haplotype. Then we can study those haplotypes. And if you have several people you're studying and they're all of Native American origin, they're going to be part of a similar haplotype. If they're part of European origin, they're going to have a different haplotype or a different DNA sequence. So it's allowed us to figure out a lot of evolutionary relationships. We've done that with not only within humans, within other species. And that's how we figured out how similar humans are to several of the chimpanzees and things like that. I can remember when I was doing some of my DNA studies and making some phylogenetic trees, it was very amazing to me how similar so much of our DNA was, not only to chimps, but also cows, pigs, just things you would never imagine. And probably the most interesting thing, in my opinion, that we can use these SNPs for is to look for inherited diseases. So if we can actually follow along a phylogenetic tree, or excuse me, a family tree, and look at who's likely to be carriers for specific diseases. And of course, the important part of that is maybe we can stop the spread of certain diseases. Okay? So I'll see you guys in class on Tuesday, and we're going to go back over what I expect you to do with this material.